Hello and welcome to Teach Me in 10. In this episode, we're joined by Nick Scales, a technical sequencing specialist at Plasmidsaurus. Nick discusses how Plasmidsaurus is transforming sequencing workflows with faster, more affordable services driven by the most powerful and innovative sequencing technologies. From whole plasmid sequencing to rapid RNA-seq analysis, he'll explain how researchers can uncover hidden construct issues, reduce turnaround times from months to days, and accelerate scientific discovery with integrated analysis workflows. Hi, Nick. How are you doing?
I'm doing great. Thanks, Laura. How are you?
Yeah, all good. Thank you. Looking forward to hearing more about Plasmidsaurus. So for those not familiar, could you start by telling us a little bit about Plasmidsaurus?
Yes, of course. So Plasmidsaurus offers DNA and RNA sequencing as a service. Our mission is to provide biological insights as fast as possible without sacrificing quality. Our founders are all scientists, and that is really important to our identity. They were all at the bench themselves once upon a time, and so they're intimately familiar with the types of problems that scientists face. They founded Plasmidsaurus with the goal of relieving those pains. We've got lots of R&D scientists, and they iterate and improve the sequencing chemistries. And on top of this, there's a big team of bioinformaticians who build really excellent pipelines, and we always include the analysis at no extra cost. So our goal is that instead of just delivering you the FASTQ files, we deliver actionable biological insights. Our goal is to take on all of the boring QC stuff to give scientists more time to focus on changing the world with their experiments.
Amazing. And the first project was whole plasmid sequencing, right?
Yeah, that's right. So back in 2016, our CEO, Mark Buddy, was trying to sequence his plasmids using the gold standard technique of Sanger sequencing. But it turns out that there are certain problems that Sanger is just unable to overcome, no matter how much you try to kind of cleverly redesign the primers over and over. So Sanger sequencing, like most sequencing platforms, relies on DNA polymerase. DNA secondary structures like hairpins are really tough for polymerase to get through. And so you essentially get no sequence data for these regions. And on top of this, Sanger sequencing can't handle like repetitive regions, high GC content, inversions. And so if your insert contains any of these kind of quote unquote problematic sequences, you just don't get any data. And on top of this, the read length of Sanger is limited. So it's usually something like 800 bases. And so even if you are able to sequence through a portion of the insert, you're usually forced to assume that sort of the rest of the backbone hasn't changed at all. And so now that we have made it extremely fast and affordable to get the whole plasmid, people tell us every day, like literally every single day, we get emails about, I finally understand why my experiment failed. I spent all this time troubleshooting, and if I'd done this, then I wouldn't have had to waste that time because I've got this giant insertion in the backbone right in the middle of the antibiotic resistance gene or whatever it is. And we've even heard from some people that they'd spent an entire year troubleshooting, and this would have saved them that time. So the important insight that Plasmidsaurus had was, what if instead of Sanger, you used Oxford nanopore sequencing on plasmid? So the nice thing about that is there's no polymerase. Instead, DNA helicase is able to sequence right through the tricky hairpins. GC content doesn't make a difference. Repetitive regions are really easy, and there's no upper read length limit. So I think the current record is something like 4 megabases, so like an entire E. coli genome in a single read. And so in summary, that approach has been really, really successful, and it's become the new gold standard. It's kind of the way everybody does plasmid sequencing now.
What discovery then? So what was next?
So we've got other products that utilize the advantages of nanopore sequencing, which hopefully I'll have time to touch on later. But something that is important to us that we always like to do when we're making new products is make sure that that product is going to have a tangible impact on the scientific community. One of the most important ways that you can assess what's happening inside a cell is to look at the messenger RNA, which transcripts are being upregulated, downregulated, and how much. And there are 2 or 3 main ways that people look at this. So RT-qPCR is great because it's really fast, and basically every lab has got a PCR machine, and so it can be done right there by you in the lab. But because you need to design primers, you need to have some a priori knowledge of what you think is going to change or what you think is going to be interesting, which kind of holds you back. And even if you do have some understanding beforehand, you obviously only get results that you have designed primers for. There are also microscopy-based methods like FISH, which are really important and produce these really lovely images. But again, you run into the a priori knowledge problem. And on top of that, they're extremely slow. Right at the other end of the sort of transcriptome-wide spectrum, RNA-seq has become really popular because of this exact problem. You don't need to have any knowledge of what you think is going to change. You get information about 15,000 genes which change in expression. Imagine trying to design 15,000 primers for qPCR. But the downside here is that RNA-seq is expensive. It's usually something like $200 a sample. It usually takes a month or two to get your data back. I've heard from some people that it can take over 9 months to get your data back. So in October, Plasmidsaurus launched our version of RNA-seq, which costs $50 and only takes 3 days. On top of that, when we were building this out, we heard that not only is it slow to actually get the data back, you also have to wait a month or so for the bioinformaticians to do your analysis. We know that bioinformaticians are almost always the busiest person at any given company because they're working on everybody's project. And so, you kind of just have to wait your turn for that. And so to solve that problem, we've built the differential gene expression right into the product. So obviously, you get the raw FASTQ files, you get the deduplicated BAM, and the count tables. But the fun part is that we've got some really nice UI on the website that is really easy to use, very intuitive. And so you get all of the QC information like heat maps, PCA plots, multi-mapping rates. But because we do the differential gene expression, you also get the volcano plots, the MA plots, the pathway enrichment. And there are features like when you hover your mouse over the pathways, the genes which are in that pathway kind of change color in the volcano plot. And so the goal here is really just to make exploring what is happening inside the cell extremely user-friendly. And so in summary, you essentially take your cells, you put them in Zymo DNA RNA shield. And the nice thing about that is then it's room temperature. And you can also send total RNA in a different buffer. It's made by Genovis. It's called SeqGuard Dino Preserve. And again, that's room temperature, which is nice. So you just drop it in the Dropbox, which is right outside your building. And then we'll do the extraction, the library prep, the sequencing, and the analysis, all for $50 and in 3 days.
$50 with a 3-day turnaround seems almost impossible. How are you able to do it so quickly and affordably?
Yeah, we hear this a lot. Well, there are multiple pieces. So firstly, we automate the extraction and the library prep. So that obviously speeds everything up and massively reduces batch variability. And secondly, because we built the library prep in-house, almost, a lot of the decisions were made with speed in mind. So it's optimized as much as possible to still give really high quality results, but while being extremely fast. That's with the big team of R&D scientists that we've got. And thirdly, because our brand is built on speed, that's one of our major things. We want to make sure that we get you those biological insights as quickly as possible. And so we don't wait to batch the perfect number of samples on the flow cell before we run. We just run them because you need your data back as fast as humanly possible. And we're here to provide that.
Lovely. So what is next on the horizon for Plasmidsaurus?
We have got 2 new products that we just very recently released. So there's genotyping and shotgun metagenomics. So genotyping uses the library prep from a premium PCR. So this is sequenced on nanopore sequencers, but it will do variant calling instead of building a consensus. So because you can create these really nice long amplicons, it becomes really easy to phase. So as diploid organisms, you've got 2 copies of most genes, 1 on each chromosome. And when you're doing gene editing, it's very important to know if the changes that you've made happen on both alleles rather than just one of them. So in an example of a knockout, if you still had 1 working copy of the gene, then usually the function doesn't change because it can kind of cover for the other one. So as part of QC, it's important to make sure that you can check that you've successfully knocked out both copies of the gene. And so with Plasmidsaurus genotyping, you use the length to phase the amplicons into alleles, which is straightforward once they get long enough. And then we do the variant calling. And again, we've got these nice kind of user-friendly plots that you can click around and kind of zoom in. You can see the per base data, and then there's a multiple sequence alignment of all the different alleles that it finds. It will kind of default to using whichever is the most abundant allele as the reference, but you're also able to upload your own reference and have them all compared to that. And so you get to see sort of whatever changes there are, if there's SNPs, indels, the sort of downstream predicted amino acid changes. For example, you get to see really easily, and it will pop out in bright red if whatever the change is introduced an early stop code on, and that's going to give you truncated proteins. kinds of things are really nice to know about. And so you submit those amplicons to us. We do the sequencing and the analysis, and it is only $30 for 3,000 reads, which is the same as the sequencing cost. So the analysis is free, and it takes 1 day. Additionally, we just launched shotgun metagenomics. So as I'm sure you know, we live in a microbial world. Essentially, every single surface on Earth has got a microbial community on it. And scientists are really only just beginning to understand the implications of all of this microbial life. For studying these communities, one of the nice things about shotgun metagenomics is that it gives you a less biased look at the microbial community in your sample compared to, say, 16S, because you're skipping that PCR step. And so this allows you to assess the bacteria, the fungi, the viruses, the protists, really whatever is in there. And we do this on Illumina. This is 2 by 150. It costs $90 for 2 gigabases, so that's 6.6 million reads, or $140 for 10 gigabases, that's 33.3 million reads. And as always, as hopefully you know by now, included is the analysis. And so for this product, we will do the taxonomic classifications and report those relative abundances. But the other reason people like shotgun metagenomics so much is that you get all of this functional information. So we will, of course, characterize those functional genes with KEGG, gene ontology. You can look for antimicrobial resistance genes. And we're still building this out, but soon it will incorporate things like diversity metrics, ordination plots, etc. So in summary, Plasmidsaurus exists to make your life easier. By regularly incorporating these kinds of QC checks into your workflows, you will save yourself so many headaches and so much time troubleshooting failed experiments, because when you sequence everything, it becomes really easy to spot what would have gone wrong. At Plasmidsaurus, we are reducing delays and delivering actionable biological insights directly to your inbox as quickly as it can possibly be done. Thank you, Laura, so much for having me.
Yeah, absolutely, Nick. And if anyone wants to learn more about Plasmidsaurus, where can they head?
Yes, please check out our website at plasmidsaurus.com.
Lovely. I have had a little look at it, and it's well worth a visit, even just for the dinosaur cartoons. They were amazing. Thank you for joining us. Take care.
Yeah, thanks so much.
Thank you for watching this episode of Teach Me in 10, brought to you by Plasmidsaurus. To learn more about Plasmidsaurus sequencing services and how rapid sequencing and integrated analysis can accelerate your research, check out the links in the description. And as always, if you enjoyed this episode, don't forget to like and subscribe to stay up to date with all of our latest releases. See you next time.